This is a short video to accompany Module 4 in the area of genetics and heredity. This video will focus on pedigree analysis and Punnett squares. In order to understand how inheritance works, we need to understand a bit about DNA, which we started in the last module, Module 3, with a basic understanding of cellular reproduction. So as we've learned up to this point, our DNA is condensed into chromosomes. And as humans, we all have 46 chromosomes. Our 46 chromosomes are inherited as a set of 23 from our mom and 23 chromosomes from our father. So when we are a fertilized egg, half of our DNA comes from mom, the other half of our DNA comes from our father, and that 23 plus 23 gives us our full complement of 46 chromosomes. Now, your chromosomes are nothing more than all of your DNA wrapped up and all coiled around itself in these very tight bundles. And so your chromosomes don't look like this normally. Normally in your cells, your chromosomes are sort of spread out in what we call chromatin, which is a thinner type of arrangement. It's not as compact. All the time in the nucleus of your cells, your DNA is wrapped around histone proteins to form these things called nucleosomes. But normally your DNA is coiled around these histones and then kind of spread out as chromatin throughout the nucleus. Whenever your cells reproduce, though, that DNA becomes tightly compacted and we form these very thick, dense chromosome structures. Now, your DNA is needed to ultimately make proteins. So let's look, if we can, here at the basic structure of DNA again, just to review. DNA is a double helix, and we have 4 what are called nucleotide bases in our DNA. A stands for adenine, C stands for cytosine, G for guanine, and T for thymine. So these A's, T's, G's, and C's are arranged in a way such that adenines pair up with thymines and cytosines pair up with guanines. So A's and T's always form pairs, and C's and G's always form pairs. And if we look at how that occurs in the structure of the molecule, the overall structure of the molecule, we have one ladder, if you will, or one half of the ladder of DNA, which will base pair with those bases in the center with the other half, or the other side of the ladder. So the bases essentially make the rungs of the ladder. And then if you could imagine taking the top and the bottom and then kind of twisting them, that's how DNA gets its double helix nature. So DNA is a double-stranded nucleic acid. The bases of our double-stranded nucleic acid are A's, T's, G's, and C's, adenines, cytosines, thymines, and guanines. And our adenines always base pair with thymines, cytosines always base pair with guanine. Last week, we talked about how the DNA molecule unwinds itself to be replicated so that the cells can replicate and each cell will get the same complement of DNA. But the important thing for us to move on to this week is more of an understanding of how DNA makes proteins. So DNA in our chromosomes contains genes, and genes are nothing more than a length of the DNA that ultimately is going to encode a protein. And in order for that protein to be produced, our DNA has to separate, and there's an enzyme called RNA polymerase that opens the DNA, and then it reads one of the strands of that nucleic acid and it copies it into a single-stranded piece of RNA. this is what we call the RNA transcript. Now, RNA in turn is going to be translated into the protein, and the protein is the functional component. Proteins are what we see. They're the components in the cells that actually do work, like enzymes or proteins, that sort of thing. So whenever we talk about DNA encoding our, or leading to the expression of our traits, what we're talking about is that DNA encodes the proteins, and the proteins are the physical embodiment of those traits. So let's talk more about that process. Here we have our chromosome, and our chromosome, as we just discussed, is condensed DNA, and the enzyme RNA polymerase, which we see here, is responsible for recognizing the start of a gene and then producing this single-stranded piece of RNA, which we call the transcript. Now let's take a look over here at how RNA becomes protein. So this is the gene at the top. That's what we're looking at here. Our genes have particular sequences of DNA that identify them as having different functions. So there are certain sequences which are associated with something that we call a promoter. Now this promoter is where that RNA polymerase enzyme has to bind. So RNA polymerase has to recognize that promoter sequence and bind to it, and then it's going to be directed to what's called a start site. And that's where RNA polymerase knows then to begin the process of transcription. And transcription is making the single-stranded piece of RNA that's the RNA transcript, and that's what we see here. Now, RNA is going to have things called exons and introns. So these correspond to the exon sequences, and then in the red boxes, the intron sequences. So introns are sequences in our messenger RNA that are going to get cut out when the exons get spliced together. So our RNA starts as a longer piece of genetic information that has introns and exons. So it starts like this as the primary transcript. And then RNA gets processed. And processing has a number of different steps, but suffice it to say that the RNA final product, the transcript that actually gets translated, is much smaller, and it does not contain these intronic sequences. They've been spliced out, and all the exons have been essentially glued together to form our final transcript, or the final product, and that's what ultimately gets translated, and translation occurs out back in the cytoplasm, out at the ribosomes. So the ribosome is the structure that actually does the translation into the protein. And the protein is these amino acids. We've talked about them before in the class too. Are amino acids that get hooked together in a sequence that is directed by the order of the nucleotide bases as they occur in the RNA transcript. And then as those amino acids get hooked together, eventually they fold up into the fully functional protein. And proteins are the things that do the jobs. They're the things that work in our cells. And so if we can come over here, there's another quick view of it. So our sequence of DNA is what we see here at the top. Again, RNA polymerase opens up that DNA and it reads one strand of it, and then it copies it into this complementary strand called RNA. RNA is just a single-stranded molecule, whereas DNA is double-stranded. Incidentally, in RNA, the base uracil replaces thymines. So everywhere where you have an A in the DNA, the corresponding complement to that in RNA is a uracil. So that's why you see the U's. RNA is single-stranded, uracil instead of thymine. Those are two of the differences between RNA and DNA. So once RNA polymerase copies that full transcript, then the transcript gets processed, introns get cut out, exons get glued together, and then translation by the ribosomes allows for the formation of the protein, the polypeptide, which is the protein, and that is the functional component. Okay? So these bases in the RNA transcript are read, if you will, or interpreted in groups of 3, which give rise to these amino acids. And so the process of translation is the process where the ribosomes essentially read or interpret the information in the RNA transcript, and then they use that information to correctly lay down a sequence of amino acids and then bond those amino acids together using what are called peptide bonds. So we go from DNA to a single-stranded RNA transcript, which gets processed, and then in the ribosomes, the sequence of the bases are interpreted in groups of 3 to give us the appropriate amino acids, which are all hooked together to make proteins. So in a very quick synopsis, that is how, in our cells, we go from DNA to messenger RNA and then ultimately to a functional protein. A nice summary of that process is here. DNA is the information that encodes the master code, and that master code has to ultimately get translated into something that's the functional component, and that's the protein, and the protein is what governs the trait. And here the trait is whether we have attached or free earlobe. So this is an attached earlobe in B, and this is a free earlobe in A. And those kind of traits are directly the result of the proteins that we express. So here's another way to look at that. DNA in the chromosome, which is going to be transcribed into mRNA and then translated into the functional protein. And again, the protein is all of those amino acids hooked together, and the proteins have certain jobs. And in this case, we're looking at a protein that would be responsible for providing the color to the iris of the eye. And so those proteins, the function of that particular protein is it provides, in this case, the green color in the eyes, right? So that is how proteins direct certain functions or traits that we see expressed in all organisms, okay? So information flows from DNA to messenger RNA, ultimately to protein, and proteins are the things that get expressed and they define our characteristics, our traits. So what we're going to spend the rest of today talking about is those traits and how we understand the inheritance of those traits. One of the most important tools for understanding inheritance and characteristics is to understand Punnett squares. If you went through the resources, there were a lot of resources and videos that talked about the things that Gregor Mendel studied in pea plants and some of the traits that he studied in pea plants. So let's start with a basic Punnett square based on a couple of characteristics. Let's start with a single characteristic that Mendel talked about with respect to his pea plants. So we will use the letter T, capital T, to designate tall plants, and a lowercase t to designate dwarf plants. And I'll put 2 of them there. Because in normal, classic Mendelian inheritance, a dominant gene, which is indicated by the presence of a capital letter, dominant genes, you only need one of them to express the dominant phenotype. To express the recessive phenotype, you have to have 2 recessive alleles. Now remember, we get 1 allele from our mom and 1 allele from our father. So let's say that we have a case like this, where what Gregor Mendel called a true breeding tall plant. True breeding talls, or true breeding dwarfs, were what we call homozygous. homozygous means that there are two of the same allele. So in this case, sorry, I can't spell and talk at the same time. In this case, we have a homozygous tall plant, which is two dominant T's, and we'll cross that by a homozygous recessive plant, which are two little t's, two recessive alleles. And so if you do this particular cross, our homozygous tall can only produce gametes. And now remember, whenever we produce gametes, eggs or sperm, those gametes only get one copy of every single allele. So we have two chromosome number 1s, we have two chromosome number 2, etc., etc. One we get from our mom, the second one we get from our father. So in the gametes, so we'll call this one the eggs, and we'll call from this plant, we'll call this the male plant, which would produce the sperm, right? So this plant can only pass on the tall allele because it has two of them. It can only pass on tall. This plant can only pass on the recessive allele. Again, it's got both recessives, so the only type of gamete that it can make, the only type of sperm in this case, is a sperm which would contain the recessive allele. So when you do this cross, true breeding tall by a true breeding dwarf plant, 100% of the offspring, and this what we're going to call the F1, the first familial generation, 100% of them will be what we call heterozygous. Heterozygous means that they have one of each, so one dominant allele and one recessive allele. Okay, so the heterozygote has one of each. Okay, if we do an F1 cross, so we take an F1 by an F1, so here we take a heterozygote by another heterozygote, then let's figure out what those offspring are going to look like. Okay, so we do this again by figuring out what the gametes are going to be. So our gametes from this parent, from the first parent, again, we'll say this is the female, so it can make two types of eggs. It can make eggs that have a dominant allele, right? Or eggs that can carry the recessive allele. Remember, we have to divide them in half, right? So each egg gets only half of the DNA, so we can either get the dominant or the recessive, right? And the same thing, this parent that's going to make the sperm has the same options to make sperm, right? One sperm can get the dominant allele, the other half of the sperm can get the recessive allele, right? Okay, so if we take these gametes and we put them in a Punnett square. So let's look at our square here. So normally we put the sperm across the top. So a dominant and a recessive. And then here we'll put the eggs on the side. Again, a dominant and a recessive. So when we look at this particular cross, the top square in the upper left quadrant here can get two dominant alleles. That's what we call homozygous dominant, right? And so the phenotype in this plant is going to be tall, right? And then this guy over here, the top right quadrant, is going to get the dominant allele from this parent and the recessive allele from the other parent. So it's a heterozygote, its phenotype, however, also is going to be tall. The bottom left quadrant gets the dominant allele from the sperm on top, the recessive allele from the egg on the side, and its phenotype also tall. And then our bottom right quadrant gets two recessive alleles, and our phenotype in this case is going to be a dwarf phenotype. Okay? So I hope you followed that. I hope you followed where those were coming from, right? So that one and then that one, right? So I hope you were able to follow where the alleles were coming from and going in in those cases. Okay, so hopefully that makes sense to you. That is what we call a single gene inheritance. So let's work on seeing if we can do one with 2 genes involved, not just one. Let me maybe do these guys in red. Uh-oh. All right, let's look at more than one trait now. We use our T for tall and recessive T is dwarf. And in this case, let's do another characteristic. Let's call it round or wrinkled seeds. So we'll use a W for round, dominant, and then a lowercase w for wrinkled. So we'll have our homozygous dominant, and we're going to cross that to our homozygous recessive, and I'll try to make my lowercase w's curvy and my dominant uppercase w's pointy so that we know the difference between dominant and recessive. Okay, so if we do this cross, homozygous dominant tall and homozygous dominant round by our dwarf wrinkled, then all of our F1s, again, 100% of them are going to look exactly the same. This plant can only pass on tall and round seed characteristics, whereas this plant can only pass on dwarf and wrinkled. So 100% of our F1s here have to get a dominant T from this one, a recessive T from the other plant, a dominant W from this plant, and a recessive W from the other. So 100% of our offspring in this cross are going to be heterozygous at both loci, and the phenotypes of these guys are going to be tall with round seeds, right? So all of them are going to be the same. If we do our F1 cross, F1 by F1, we take tall, heterozygous tall by heterozygous round, and we cross that with the exact same thing, right? And so in this case, if we figure out what our offspring look like in this case, that's what we're going to use this Punnett square for. So let's figure out what the gametes are like from these guys, right? Eggs and sperm. If this plant is making the eggs, so this is going to be our female plant, this is going to be our male plant over here. So the gametes here, the eggs from this plant are going to have a couple of possibilities, right? We can get, let me erase these and maybe make this a little bit easier to follow. Oh, I can't erase that. All right. So let's go back here. All right, if we look at our eggs, the production of our eggs, we can sort this T with this W, right? So an egg that will get a tall and a round allele. And then down here, we're going to sort this T with the lowercase w. So another egg that maybe gets the tall allele with the wrinkled seed allele. And then this recessive T with the dominant W. So another egg, whoopsie, sorry about that. In this case, we get the recessive T, so little t, dominant W, and then the little t with the recessive W here, so little t and little w, right? So now we have 4 possibilities of our gametes, and those possibilities are all completely independent of one another. It's just a matter of how these different alleles assort with one another, and there are 4 possible combinations that we can come up with. So the gametes from one parent are going to be these 4 different types, and the other parent is going to be able to produce the exact same set of gametes. Okay, go back and watch that again if you didn't understand exactly how those assort. And again, I'll refer you to the resources because there's a Mr. Anderson video in your resources that really goes through that same process really very well. So now let's go over here and let's look at how we build a Punnett square now for 2 characteristics. All right, so here we have our sperm across the top, and our sperm will have the same possible combinations. The tall allele with the round seed allele, the tall allele with the wrinkled seed, the dwarf allele with the round seed allele, and dwarf with wrinkled. And then our eggs, again, the same 4 possible gametes. So I will put them on the other side. Okay, hopefully you guys can differentiate my dominant W's from my recessive W's. All right, now let's go through and figure out what the combinations of offspring can look like. So this offspring gets 2 dominant alleles, so 2 dominant T's, a dominant T, and a dominant T, right? And then again, here we have the dominant W and the dominant W. So we get, I should make this a little bit closer. So we get something that looks like this. So this particular plant gets 2 dominant alleles for tall, 2 dominant alleles for round seeds. And so this particular plant is going to be tall with round seeds. Okay, so I'm going to fill in the rest of the chart, and then we'll come back and talk about them. Now that I have completed the rest of the Punnett square, let's go back and look and see if we can figure out the phenotypes. So we use tall, round. We're just going to abbreviate with a capital or with a T and an R for all of our tall, round plants. So these are plants that are tall and they produce round seeds. So let's count how many we get. I'll designate these guys. I'll keep them in red because that's what we started with. So this guy is one of them, tall, round. So this guy is going to produce tall, round. Maybe I shouldn't use abbreviations like that. That might be confusing. Let's do stars. So all of our tall, round plants, we'll give them stars, right? So all 4 of those guys are going to be tall and produce round seeds. And all of these guys, the same thing. And all of these guys, you'll notice, they have to have at least 1 dominant T allele and 1 dominant W allele. Okay, so dominant T, 1 dominant T, 1 dominant W at least, okay? So if we count those guys up, we should have 9 of them. So 4, 5, 6, 7, 8, 9. So 9 of our offspring in this cross. So we'll have 9 that are tall plants and they produce round seeds, okay? And then I'll use a different color for those guys that are going to be tall but make wrinkled seeds. So they'll have at least 1 dominant T and they'll be homozygous recessive for the wrinkled characteristics. And those guys will have the blue circles in them. So we're going to have 3 that will be tall plants, but they'll make wrinkled seeds. The plants that are going to be dwarf plants but make round seeds, I'll indicate with a green triangle. So these are all homozygous recessive at the T allele, and they have at least 1 dominant W allele. And notice we have 3 of them. They're going to be dwarf, and they will make round seeds. And then we will have just 1 plant, and I'll indicate that guy right here, and it is going to be dwarf and make wrinkled seeds. So we get 1 dwarf and wrinkled seeds. All right, so this is what we call a 9:3 to 3:1 phenotypic ratio. And so our phenotype is the tall with round seeds or wrinkled seeds or dwarf. Those are all phenotypes. They're expressions of our genes. And so across, like this one, where we have 2 heterozygotes, so indicate them here. So this is what's called a dihybrid. So 2 heterozygotes is what we call a dihybrid cross. And a dihybrid cross will result in an offspring of a ratio of offspring of 9 to 3 to 3 to 1. So our two dominant characteristics will have 9 of those. 3 of them will have one of the dominants and the other recessive characteristic, and then 3 will be the opposite. The other dominant characteristic with a recessive, and 1 will be recessive at both loci. So this is a pretty classic standard what's called dihybrid cross. And again, if you need further explanation of this, if you check out the resources folder, I have a bunch of videos, maybe not a bunch, but I have several videos that will help you understand that, the nature of a dihybrid cross as well. To summarize some of the things that we talked about with Punnett squares, it is important that you understand the difference between homozygous and heterozygous. So homozygous is having two alleles, two of the same alleles. So in this case, two dominant alleles, that's homozygous dominant, and two recessive alleles, that's homozygous recessive. The heterozygote has one dominant and one recessive allele. So hetero, meaning different, has two different alleles. When we talk about multiple traits, so we can talk about homozygous dominant for tall, homozygous dominant for production of round seeds, or we could talk about heterozygous at both loci, right? So heterozygous at the locus for tall and heterozygous at the locus for producing round or wrinkled seeds, okay? And it's inherent then that you understand the difference between dominant and recessive. The dominant allele is going to be expressed over the recessive allele, which means that the heterozygote condition, so this guy, the heterozygote, that's the genotype, the two alleles, that's the genotype. So the phenotype of the heterozygote is going to be dominant. The phenotype of the homozygous recessive is going to be recessive. In this case, that recessive, in this case, is dwarf. Okay, so genotype is what do the genes say? So this is the combination of the alleles, right? The phenotype is the expression of those. So the genotype would be homozygous dominant or heterozygous. The expression would be tall or dwarf or round or wrinkled. That's the phenotype. Okay, so that's just a quick summary of the things that you guys should understand if you understand the unit on Punnett squares. There are a number of reasons to have you guys learn about Punnett squares. So who doesn't love puppies, right? And if you look at this image, this photograph, it's got a black lab here, and she's got a bunch of different puppies. Half of them are black and half of them are yellow. So a question might be, well, how does a mom who's a black lab have yellow pups? And you might say, well, clearly that's from the dad. But the dad in this case was also a black lab. So how do two black labs end up having yellow pups? Well, if you understand the nature of pedigrees and you understand some of the things that we've been talking about in this lesson, you can kind of begin to figure it out. Now, this problem is a little bit more complicated because it has, it's an example, color coat in labs has something called epistasis in it. And epistasis occurs when one gene or one allele sort of overrides another allele. So in this case, there is a protein that's involved in depositing the color of the fur. So the pigment that causes black labs and brown labs to be black or brown has to be deposited in the shaft of the hairs. And so there's a second gene in this case, which is designated by the letter E, which comes into play. So if we look at two black labs that have puppies, and again, we'll look at the sperm on the top of this Punnett square and the eggs along the side. So sperm from the male parent designated on the top, eggs from the female parent here on the side. So these two parents are both black labs, but they're both heterozygous at both loci. So the black labs, they're black, so they have a dominant B allele, but both of these parents are heterozygous, so they're dominant B, little b. They're also heterozygous at the second locus, which is the one that allows for the pigment to be deposited, and that's the E locus. So they're dominant E, recessive E. So big B, big E, little e. And if we look at how this particular cross works out, from these two parents, These are all the possible types of offspring that they can have. And as long as they have at least one dominant B and at least one dominant E, then all of these guys will be black. So you'll see 9 possibilities here where they wind up with black lab offspring. If the offspring get both recessive B alleles, so little b, and at least one of the dominant E alleles, then they will be what we call chocolate labs or brown labs, and there are 3 possibilities there. However, all the dogs that get the 2 recessive alleles at the E locus, so little e, there are 4 possibilities where you wind up with little e. All of those dogs will be yellow labs. Doesn't matter what the B locus is. Dominant recessive doesn't make any difference, because if they're homozygous recessive at the second locus, the E locus, they won't be able to deposit any pigment in the shaft of the hairs. So those labs will all be yellow. And this is a condition called epistasis, and this is the reason that Labrador retrievers have 3 possible coat colors: black labs, chocolate labs, and yellow labs. And it's another reason why it's important for you guys to understand Punnett squares, because if you were going to buy a lab or if you were breeding labs, then it's important to understand how inheritance works and be able to predict how inheritance would work in those dogs. All right, this comes into play in lots of different characteristics, not just dogs and not just coat color characteristics, but we see it in lots of different everyday situations. So again, this is why I think it's important, one of the reasons why I think it's important that you guys have an understanding of this type of science and to understand inheritance and to be able to perform and to interpret pedigree and Punnett squares. One of the other components of this module is to understand how we do pedigrees, or some basic pedigree analysis. So in the resources, and I'll go over them quickly here, these are some of the symbols that are used commonly in the production of pedigrees. So pedigrees are charts kind of like this guy over here, where you can visually lay out inheritance patterns of a particular family or group of families with respect to a given trait or traits. So some of the common symbols used: squares are used for males and circles for females. A marriage is indicated by a horizontal line connecting individuals. What's called consanguineous, meaning relationships between closely related individuals, are indicated by 2 lines, a double line connecting the male and the female together. So twins are indicated by 2 lines that come off of the same sibling line. So identical twins are connected by a horizontal line between them, and fraternal twins lack that horizontal line. If an individual is affected by the condition that the pedigree is tracing, then they're filled in symbols, completely filled. Carriers for a particular condition are partially filled in. Oftentimes you just see them like this with a dot in the center of their symbol. Affected by the history, sometimes you get symbols like this that are hatched, and these individuals are usually affected by history. Again, female circles, males are squares. And then if an individual in a pedigree is deceased, there's a line diagonally through the symbol. If there's an individual of unknown sex, like a pregnancy, sex is undetermined, then they're given by a diamond shape. And so we use Roman numerals to identify the generations, and this would represent generation 1, so parents, and then dropping down from the parent line is a sibling line. And so these 3 shapes represent 3 children of this relationship, 2 boys and a girl in the middle. Okay, so and 1, 2, and 3 designate the order of those particular siblings. Okay, now in any generation, every individual in that generation oftentimes is going to be designated with a number. Sometimes those numbers start at the beginning, and then every single individual represented in the pedigree is numbered consecutively from the earliest generation of grandparents or great-grandparents. So let's take a look at an example pedigree over here. I'll pull that in. So here we're looking at two parents. So here we have parents 1 and 2, and these parents have had 4 offspring, 4 children, 3 daughters and 1 son. And then the relationships between these individual offspring. So this daughter married this individual male, and then they had 2 children. This daughter married this male, they had 2 children, so on and so forth. So you can follow through the generations of the pedigree that way. Those who are affected by the particular condition that this pedigree is tracing are indicated by the filled squares, and those that are affected by history are indicated by the hatched marks. Here are a couple of example pedigree charts to give you an idea of how to interpret them and what they mean. The first one that we'll talk about is colorblindness, which is inherited in a manner called X-linked. Now, colorblindness, the gene for colorblindness occurs on the X chromosome, and because females are XX, so I'll put it down here, females are XX, boys are XY, because males only have one X chromosome, they're the ones that normally are affected. Females are only affected if they get both defective X alleles, which does happen, but it's pretty rare. So you normally see X-linked conditions showing up in the males, again, because they only have one X chromosome. So in this case, if they get the colorblind allele, they're going to be colorblind. If a female gets the colorblind allele but one normal allele, she'll have normal color vision. So let's look at the colorblindness pedigree. Here we have the female, the first individual in this family who is the carrier. She's the initial carrier. She has 4 children. One of her defective alleles, so we'll give her genotype here, right? She's one normal allele and one colorblind allele, and Papa over here is a normal XY. So this daughter inherited that colorblind allele, so she's what we call a carrier. The son over here, he got the colorblind allele. So number 5, this son is the colorblind X and a normal Y. So this son happens to be affected. He's colorblind. This daughter right here, she's a carrier. So let's look at the first family as we go. So number 2, the daughter right here has a normal husband. They go on to have 3 children. Her son is normal. So he's a normal XY. He received her normal allele. And this daughter is a normal XX. She received the normal X from dad and the normal X from mom. But this daughter got the colorblind X from mom and then the normal X. So daughter number 3 in this generation marries individual number 4, and they go on to have several children as well, 5 kids. This son, number 1, gets the colorblind X and then a normal Y chromosome. So he's colorblind. 4 and 5, these sons are also colorblind X with a normal Y. So all 3 of those sons are colorblind. Number 3 got the normal X and the normal Y, so he's not affected. And the daughter number 2, she's a normal XX. Okay? You can see in this generation, these 2 sons are both normal because they had to get their Y chromosome from their dad. So in this case, mom is a normal XX, so she gives these 2 sons her normal X. That's all she's got, so one of her normal X's. And these sons have to get their Y chromosome from their father. So these 2 sons are perfectly normal. In fact, with a normal mother who's not a carrier, they have to be normal because they have to get the Y chromosome from their dad. So anytime that there's a father in a family like this who is colorblind, he cannot pass the colorblind allele to his offspring because it's an X-linked characteristic. To his male offspring, because it's an X-linked characteristic. Okay, so the boys have to get the defective X from their mom, not from their dad. right? Fathers can only pass a defective X on to their daughters. All right, so let's take a look at some other types of inheritance here. This is another X-linked characteristic. It's called Duchenne muscular dystrophy. And in this case, again, we have the mom who's a carrier passes that defective X onto a son. This son is deceased. She passed one of her defective X's onto a daughter as well. This daughter passed it on to both of her sons and to one of her daughters. This daughter passed it on to one of her sons here and one of her daughters indicated here. And again, this daughter passed that defective X on to her son. Notice all of the female offspring are just carriers because they have a normal X to compensate. The male offspring that inherit that defective X chromosome are all but one of them. This youngest one is still alive. The rest of them are all deceased. Okay, so classic X-linked inheritance where females are carriers, males are affected. Okay, and then let's look at a couple of other types of pedigree charts. This is a pedigree chart for a condition called PKU, phenylketonuria. PKU is a condition where those who are affected have the inability to process certain amino acids, and that amino acid is phenylalanine. It tends to build up in the system and causes some neurological complications. Notice in this pedigree, first off, we have affected males and females both. So it's not likely to be X-linked, and we can go back and confirm that. In this case, we have two parents, both of whom are carriers. In this pedigree, too, carriers are indicated by the hatched pattern, which is a little different than what we've seen in the past. Different pedigrees do it different ways. So our carriers are indicated by the hatched pattern. So both of these parents were carriers, and of all of the offspring that they had, 1, 2, 3, 4, 5 offspring, 2 are affected, and then these 2 are carriers. And so this is a classic autosomal recessive inheritance where you have to have both recessive alleles to express the condition. So let's say we'll use the PK and U. None of them make great lowercase and uppercase letters. So we'll use the lowercase or uppercase P for PKU, right? So our father here would be a heterozygote. Our mother, the same thing, big P, little p. I'll try to make my lowercase p's look different. This son would get the lowercase p from both parents, so he's affected. This daughter is not a carrier, so presumably has the dominant p's from both parents, right? The dominant alleles. This daughter is affected, so she's got lowercase. This son is a carrier, so he's one dominant, one lowercase, and this daughter, the same thing, carrier, so one dominant allele, one recessive allele, right? So this the second son has married another woman, and she is not listed as a carrier, so presumably homozygous dominant for normal. And then they have passed on homozygous dominant for that daughter, homozygous dominant for that son, and then here this offspring would be a heterozygote carrier. And then this affected daughter here marries a normal husband, and they have 2 children, a son who's a carrier and a daughter who appears to be perfectly normal. Now, if we look at this particular pattern, I want you to notice if the daughter in this case appears to be normal, but mother is affected and this is a recessive condition, can this occur? The answer to that question is no. This daughter cannot be perfectly normal. This daughter has to be a carrier. And the reason is because mother only has recessive alleles to pass on. So even though that is not a shaded symbol to indicate that it's a carrier, she must be a carrier because the mother only has 2 recessive alleles to pass on. That's all mom has. So all of her offspring have to inherit a recessive allele. Okay? So even though that daughter is not shaded, she absolutely must be a carrier as well, right? Because Pop here has dominant alleles to pass on, so he will pass on one of his dominant alleles to that daughter, but Mom only has recessive alleles, so one of those recessive alleles has to be passed on to that daughter as well, okay? So she must be a carrier. All right, I wanted to point that out to you in that pedigree because that symbol is not shaded, but it must be shaded. There's no other option there. She has to be a carrier. And then our last pedigree that we're going to talk about, this is kind of an interesting one because it's got a couple of things going on. The condition in this one is called CMT, and it's a neurological condition. It's got a weird, funny name, but it's a neurological condition that causes the breakdown of normal neuromuscular function, so people become impaired in their ability to walk and in certain motor skills. So one of the interesting things about this one is the consanguineous relationship here between these two parents. So the double line between these two parents indicates that this is a consanguineous relationship. The journal article for this one I'll put at the end of the presentation. These so the father in this case is and the woman in this case she's an aunt, so the relationship between these guys is a familial relationship, so they are it's consanguineous, so they are indicated with a double line there. So if we take a look at the offspring in this case, both mom and dad are carriers for this condition, which is called CMT. The proband is indicated, and this is the first pedigree that had a proband, and that's the P with an arrow pointing to the particular symbol. That means the proband is the individual that is the purpose for initiating the pedigree. So this individual, this 15-year-old, the 15Y indicates that this is a 15-year-old male offspring, and this is the person who would have presented at a doctor's office or for genetic counseling, and this is the reason that the genetic tests were done. Okay, so the proband is indicated by the P with an arrow, and that means that that's the person that started or initiated the formation of the pedigree. Okay, so this son has a sister who has the same condition, CMT. She's 17, and then he has 3 other siblings, one other sister and 2 brothers that are healthy, and they range from 2 years old up to 13 years old. Okay, so in this particular condition, again, you see both parents are carriers, and both of those parents, or because they're related, in consanguineous relationships, you often see recessive conditions creep up at much higher frequency. And this CMT is a recessive condition, and you see it in 2 of their offspring. One's a female and one's a male. Okay, so it's not X-linked, it's not Y-linked. Y-linked characteristics are super rare. This is just a standard recessive kind of inheritance pattern. Okay, but I wanted to point that one out to you because it had some interesting things associated with it. So if you're asking yourself, Frost, why are you making me learn this? I just don't care. Well, this is why I'm making you learn it. Pedigrees are important for you to understand because there's a real possibility that maybe pedigrees become part of your life. There are so many examples now where people are having genetic testing done through 23andMe or Ancestry.com, and there are huge numbers of companies that are sprung up in the last 4 or 5 years that are all doing genetic testing for different things, not only for your ancestry, but also for health screenings. And this is a pedigree looking at a particular mutation in a cancer gene called BRCA. BRCA1 and BRCA2 are 2 genes that are highly associated with the development of breast cancer and other types of cancers as well. So this is a BRCA1 mutation. The designation of that mutation you can see here, 5073A to T variant. So this is an adenine to thymine variant in the normal allele. So this is a mutant allele, and they're tracing this mutant allele over a couple of generations. In this particular family, the mother of this particular family died of ovarian cancer, as did her sister. Mom died at 61, her sister died at 71. The proband of this of this pedigree is indicated here, again with the arrow. She's a 53-year-old female, and she has been diagnosed as having this particular variant, this mutation. Her her brother, in this case, also has the mutation. He is a carrier of the mutation. And then we're looking at their children who have not yet been screened for the mutation because they're obviously, according to the pedigree, not screened because of their young age. But this female must have presented for genetic testing because she has ovarian cancer, like her mother and her aunt both had. So she's presented for genetic testing to figure out if there is a reason, an inheritance pattern that is the reason for her development of ovarian cancer. Males don't have ovaries, so clearly they can't have ovarian cancer, but her brother is a carrier of the same mutation, and there are certain health effects that can happen in males as a result of BRCA mutations too. So BRCA mutations, although males don't have ovaries, they can't have ovarian cancer, they don't have cervixes, they can't have cervical cancer, they do still have the possibility of developing breast cancer and various other cancers too. So this is why it's important for you guys to understand pedigrees. It's important for you guys to understand inheritance patterns, because stuff like this is going to happen at way higher frequencies now that genetic testing is becoming so much more common than it used to be. And I think it's really important for everybody to have a basic understanding of inheritance, and you really should be able to read and understand a basic pedigree analysis, because it's going to be more and more a part of, and I hate to say it, but of everyday life, that these kind of genetic conditions are going to be discussed and researched more, and many of us may actually be seeing some visual outline of heritable diseases or conditions because of genetic screening that's going on at a much higher rate now than really at any time in history. The BRCA mutation pedigree that I just showed you came out in this particular report back in 2018, so it's only a couple of years old. But this report outlines the inheritance pattern in a couple of families, but of this particular variant that's associated with hereditary ovarian cancer. Here's the pedigree that I showed you in the previous slide, outlining the ovarian cancer and the particular BRCA variant that you see in this family. This outlines their approach to how they did the genetic sequencing, the particular area of the chromosome, and the location of the variant that they're talking about. And then some of the consequences of that variant, a changed splice site and the introduction of a premature stop codon, which alters the protein significantly and is likely associated with the development of cancer that's seen in the inheritance pattern. So if you're wondering why I make you study this, it's because I think that everybody needs to have a basic understanding of how genetics works and how inheritance works. Not asking you to learn molecular genetics at an extraordinary depth. I'm just asking you to have a basic understanding of inheritance patterns and a basic understanding of how we study inheritance. And I think it's important because genetic testing is becoming so much more common, and maybe at some point in your life you will need to know, or maybe you'll be able to help somebody else who has a genetic test and finds out that there's some weird anomaly. Maybe you can help them understand how that works. Either way, I think that it's important that everybody has a basic understanding of these concepts. And so that's why I think it's important that you guys study Mendelian genetics and have some idea of how heredity works in humans. A quick summary of what we covered in this particular presentation then. We talked about how DNA is transcribed into a messenger RNA, and that messenger RNA ultimately is translated into proteins. We also discussed the fact that your chromosomes and the alleles that you have on your chromosomes for different genes are what we call the genotype, whereas the phenotype is the expression of those alleles in the form of proteins. So genotypes are the combinations of the genes that you have, or the combination of the alleles. Alleles are the dominant or the recessive version of the gene, whereas the phenotype is the expression of that gene, the protein itself. We talked about how we use Punnett squares to predict outcomes from two different parents. We talked about how we use pedigree charts to understand inheritance patterns, and we talked about why understanding inheritance patterns is so important. So hopefully this helps you understand some of the information for Module 4. If you have any questions, please let me know. I'm hoping that these little videos are beneficial for you.